Hello everyone, my name is Farhan and we today will analyze two vital components, biglucan in Saccharomyces cerevisiae and glycinin in soybean. Let's start with yeast character. Is unicellular eukaryotic fungus measuring 5 to 10 nanometer and typically found in sugar-rich environment. Yeast reproduce asexually via budding and hold a grass status, making it highly safe for food industry and live bakery supplements. So the next material in Fauzi.
So the next materials is about the beta-glucan structures. So beta-glucan is the major of polysaccharides commons of the cell's wall of Saccharomyces cerevisiae. It's composed of D-glucose monomers linked to beta 1 till 3 glucosidic acid and bonds of beta 1 till 6 glucosidic branch. The branches structures forms a strong three-dimensions network that provides rigid mechanical strength and protections to the yeast. So the functions of this is maintains the cell and a cell wall integrity, provide the structures and also support the cell wall.
The physical properties of beta-glucan. First, solubility atau kelarutan. Its solubility is influenced by molecular weight, degree of polymerization, and chain structure. Next, viscosity atau viskositas. Viscosity increases as the molecular weight and concentration of beta-glucan in the solution increase. Next, water holding capacity atau daya ikat air. Beta-glucan is a hydrophilic yang banyak mengandung gugus hidroksil atau OH- sehingga mampu menyerap dan mempertahankan air dalam jumlah besar.
Sifat kimia biomolekul. Beta-glucan adalah polisakarida dengan tiga sifat kimia utama. Yang pertama, struktur ikatan serial yang bernilai ikatan beta-glucan 1-3 dan beta-glucan 1-4, dan sedangkan itu jamur memiliki cabang beta-glucan 1-6. Yang kedua itu kelarutan dan viskositas. Beta-glucan larut dalam air dan membentuk larutan kental. Dan yang ketiga adalah aktivitas biologis yang bersifat imunomodulator dan penurun kolesterol serta juga untuk penurun gula darah.
Back with me Farhan to answer the fifth part. The biological function of biglucan in yeast includes cell integrity as skeletal component comprising in 30 and to 60 of the cell wall, osmotic pressure to prevent cell lysis, and morphogenesis to assist cell separation during budding.
Next one is osmotic pressure, protect the cells from lysis and also morphogenesis. It's involved in the process of daughter cells separating. And the applications of beta-glucans is first in the food industry, in pharmacies, also in cosmetic and skincare, and also in animal feed.
Next, laboratory analysis method of beta-glucan. The method used to quantify beta-glucan is the ALAC 995.16 enzymatic spectrophotometric method, which employs glycanase and beta-glucosidase enzyme. The concentration of beta-glucan is calculated based on the amount of glucose formed.
So, the case study in scientific article. Jadi ada sebuah uji klinis tahun 2020 yang melibatkan 80 pasien dengan pilek yang menunjukkan bahwa suplementasi 250 mg per hari beta-glucan yang berasal dari dinding sel ragi Saccharomyces cerevisiae dan selama 6 bulan secara signifikan ini mengurangi insiden pilek sebesar 68% dan mempersingkat durasi gejala sebanyak 3 hari dibandingkan dengan placebo.
Next is about the characteristics of the glycans max in soybeans. So glycans max is a soybeans is a leguminous plant belonging to the family of Fabaceae. It's widely classified as in many countries as an important source of plant protein and oil. Soybeans are obtained from the seeds of the soybeans plant, which are harvested when mature and dried to a storage of processing. So soybeans are typically round and also oval shaped and have many colors like yellow, green, browns, or black depending on the variety. due to their high protein content and balanced amino acids. Soybeans are widely used to food products and animal feeds and also some in industrial application.
Structure of glycinin. Glycinin is the major protein in soybeans and belongs to the 11S globulin group. Its monomeric structure consists of one acidic chain plus minus 35 to 42 kilodalton and one basic chain plus minus 20 kilodalton, linked by disulfide bonds S to S, and is derived from a single precursor protein. In its natural form, glycinin is organized as a hexamer or 6 subunits. The structural stability of glycinin is maintained by several chemical bonds, namely disulfide bond, hydrogen bonds, hydrophobic interaction, and electrostatic interaction. This picture called this glycinin A chain F3 yang terdiri dari 52 asam amino dengan massa molekul sekitar 5.698,5 Dalton.
Moving on soybean, the physical property of glycinin, a massive isomer globulin protein of 320 and to 380. So maximum precipitation edge is isoelectric point of pH 4.5 to 4.8. Exhibit hecthermal stability with a denaturation temperature of 85 to 95. This physical trait give excellent gelling and texturization property for tofu or plant-based meat.
Next is about the chemical properties of the glycinin. So glycinins are right in the chemical structures of C2H3NO2 is the simplest amino acids that exists in aromatic properties because it contains both an amino group H NH2 and also the carboxylic group, the COOH. Its aqueous solutions. It commonly exists in solutions with positive and also negative characters charge on the some molecules.
So the biological function of glycinin was firstly for antibacterial and lower cholesterol, antioxidant, as inhibitor, and antithrombotic.
Next, application of glycinin. First, for food industry yang berperan dalam industri pangan. Next, natural food preservative sebagai pengawet alami pangan. Next, bio-based adhesive sebagai perekat berbasis hayati. And last, biodegradable food packaging.
The methods of analytical laboratory. The most reliable analytical methods for identifying glycinin is the sandwich enzyme linked in immunosorbent assay or ELISA. This method employs specific monoclonal antibodies. There are two key antibodies used, Z73 as the capture antibody and 4G4-HERP as the detection antibody. These two antibodies work together to form a sandwich complex with the target proteins, which is glycinin. So what makes the exceptional is the performance. It was an extremely high detection limit of only 28.75 nanogram per milliliter. This means it can detect even tiny amount of glycinin in a sample. Furthermore, this methods doesn't cross a river with other soy allergic proteins such as beta-glucan.
Let's see the case study from the paper modification of soy glycinin to improve functional property in food industry. So the modifying glycinin through lactic acid bacteria fermentation, the Maillard reaction successfully induce protein unfolding. This effectively increase surface hydrophobicity, maximize antioxidant activity, and enhance emulsion stability in food lipid system.